Kemudian dari kilometer juga sudah sama-sama kita ikuti pengenalan seperti apa hasilnya kurang lebih.
Sampai di warung jambu. Warung jambu mana? Makanya warung jambu. Ya, Mas. Jambu.
Good morning, ladies and gentlemen. Assalamualaikum warahmatullahi wabarakatuh. It is a great pleasure to welcome you to webinar CRYO-EM Laboratory Series 2, Application CRYO-EM in Health Sciences, held by Directorate of Laboratory Management, Research Facilities and Science and Technology Park, National Research and Innovation Agency BRIN, Thursday, April 18, 2024. In this occasion, we are honored to welcome our speakers, Professor Chatchai Tayapiwatana, Professor Division of Clinical Immunology, Chiang Mai University, Thailand. Good morning. Dr. Yudi Nugraha, researcher from Ekman Research Center for Molecular Biology, BRIN. And our moderator for today, Dr. Dede Heri Yulianto, researcher from Research Center for Applied Microbiology, BRIN. Ladies and gentlemen, next agenda is singing the national anthem, Indonesia Raya. Ladies and gentlemen, before we start the agenda, we will have a photo session first. To all participants, please activate your camera and for the committee to take the screenshot. Okay, I will start count from first slide. Ready? 1, 2, 3. Second slide. 1, 2, 3. Third slide. 1, 2, 3. Thank you. And I would like to remind to all participants, please fill the attendance form from the link provided by the committee in the chat feature. Now we are going to start our discussion for today, which will be led by Dr. Dede Heri Yulianto. To Dr. Dede, time is yours.
Thank you very much, Ms. Andalusi, and ladies and gentlemen, welcome to this webinar series 2 seconds for the CRYO-EM. And today, we are very thank you to Professor Cacai Tayapiwatana as the first speaker, and also Dr. Yudi Nugraha as the second speaker today. Today, we have two speakers. First will be delivered by Professor Cacai Tayapiwatana. And before we start, I would like to read his CV and allow me to share his CV. Please wait. Okay, I hope this can see to all the. So our first speaker is Professor Cacai Tayapiwatana. He's a full professor from Department of Medical Technology, Faculty of Associated Medical Science, Chiang Mai University, Thailand. And okay, and his education is from PhD from Chiang Mai University, and he already obtained so many awards during his career, and his visiting researcher is during 2022 and until 2010 in many universities, mostly in the USA, and he has published so many papers regarding the medical science. And the last, his research interest is he also has many patents from US patent, also WIPO and the Japan patent regarding the immunochromatographic method. And his research interest, I think this is very interesting to our audience if you want to make a collaboration research with him regarding the protein engineering, for example, antibody and also anchoring and zinc finger, molecular modeling and simulation, gene therapy for HIV virus, and also development of point of care immunological assay and biosensors. So today, the Professor Cacai will deliver the title Engineering Antibodies and Scaffold for the Next Generation Therapeutic. So Professor Cacai, you have 40 minutes for the presentation. And then we next continue to Dr. Yudi for the second speaker. And then we will have the Q&A in the last session. So for Professor Cacai, I will give you time to present your presentation. Please, Professor Cacai.
Thank you very much for your kind introduction, Professor Dede. So I'm going to share my screen now. So can you see my slide?
Yeah. It's clear.
So let's start. So I'm from Chiang Mai University, which is in the northern part of Thailand. I am leader of the Center of Biomolecular Therapy and Diagnostic Group. And today I am going to share my research idea and also the experience about 20 years in engineering antibodies and scaffolds for next generation therapeutics. So as you may know, antibody is one of the 3 pillars for cancer therapy. And the antibody therapy is very important due to its specificity. And this drives the demands of the antibody for the clinical usage. And of course, it costs a lot of money. In 2023, the market of the market value of the antibody therapy is about $7 billion. So it's very interesting to discover a new antibody, a new target for cancer therapy. And let me introduce you a little bit or revise a little bit about the structure of the antibody. The antibody has 4 chains, and the most important domain of the antibody is in the variable domains of the heavy chain and light chain, VL and VH. They combine and form the antigen binding site. If you look deeper into the structure of the antibody, there are 3 separate regions named complementary determinant regions on both heavy and light chains. These 3 areas or 3 regions define the specificity of the antibody. So we are going to focus only on the variable domain for this lecture. The other domain, maybe the next lecture, if you like me to share my research idea. So today we just only focus on these domains. So if you look through the 3D structure of the antibody, the CDR domain or CDR region 1, 2, and 3 on heavy and light chain, they conform the very special characteristic, or we call it as canonical shape of the antibody. The CDR 1, 2, 3 of yellow and green come close together and form the antigen binding site. So to engineer the antibody to increase the specificity or increase the affinity, we focus on engineering these 3 regions. The story of the antibody, the rising day of the antibody comes in 1935 after the discovery of the hybridoma technology by Kohler and Milstein. I started to learn the hybridoma technique when I was a master student in 1988. So this technology is very popular on that day to produce the antibody from monoclone of the hybridoma. So people develop a lot of diagnostic kits. And from the hybridoma technology, after getting the mouse antibody, they think about not only the diagnostic kit, but also to generate the new therapeutic way. But the mouse antibody is not proper for the therapeutic usage in human because it's foreign. Later, in the dawn of the genetic engineering, the scientists try to modify the structure of the mouse antibody by cloning the variable domain of the mouse antibody to the constant domain of the antibody, human antibody. So this can reduce the mouse domain and it's less immunogenic in human. But anyway, the chimeric antibody is not good enough. Later, they developed the technique to graft only the CDR that define the specificity of the antibody to the framework of human. So now the mouse structures or mouse peptide is lesser comparing to the chimeric antibody. So it's more proper for therapeutic usage. And humanized antibody, humanized monoclonal antibody the market is about 80% nowadays. They use this technology to construct the therapeutic antibody. Anyway, there are other technology to produce fully human antibody, but it's quite costly. So to produce human monoclonal antibody, in my lab, we don't have this technology yet. We just only have this technology, humanized monoclonal antibody. Because if you want to go for the, let's say for the humanized mouse or the transgenic mouse to produce the human antibody, one single mouse costs you about 10,000 US dollar for just only single mouse immunization and gain the B cell of the mouse to perform hybridoma for producing human monoclonal antibody. So it's not cost effective yet. That's why we still focus on humanized monoclonal antibody. So this is the diagram to show you that from murine monoclonal antibody, from the time of Kohler and Milstein, the human protein is just 0%. And once they introduce the chimeric technology, it's more human. And humanized antibody, it's even more. Now we have about 90% human, just only the CDR from mouse left over. And the newest technology, you can produce the fully human antibody. So as I told you, the humanized antibody is still in the market 80%. So it's worth to go with humanized antibody because people report a number of antibody that's formerly produced in mouse hybridoma. So if we have the sequence of mouse hybridoma, we can engineer it and directly generate the therapeutic antibody using humanized technology, humanization technology. So this is how it works. To have the mouse antibody, if you know the sequence of the variable domain, and you can define the CDR region by bioinformatics, then you can graph only the CDR from mouse to the human framework, variable framework. Now you get the ready to go humanized antibody for therapeutic usage. But this is by saying, actually it's harder than
มีใครฟังอาจารย์ไหม
saying.
คะมีใครเข้าฟังอาจารย์ไหม
So the process of humanization is starting from cloning and express mouse single chain variable fragment, or
ซูมค่ะ
for short is SCF. Force ปิดไมค์ด้วย Force
Sometimes parents look
Force
the
turn
wrong
off
way
your
in.
mic, your mic.
Explain makes really others.
So the process of humanization starting from cloning and expression of mouse SCFV from hybridoma cells, and then build the conformational structures by using, formerly they use homology modeling to get the conformational structure of the variable fragment of the antibody, heavy chain and light chain. But nowadays, people use AI to make the conformational structure of the single chain FV of mouse. The best one, from my knowledge, is A-body builder 2. And after you successfully build the structure of the single chain FV, then you can go to search for the proper human framework from the database of 3D structure that's already published. Human single chain FV that mostly match with your conformational structure of mouse single chain FV, then you can graph the mouse CDR to the framework of human. And then again, building the conformational structures of the humanized single chain FV and compare it against the mouse single chain FV. So why we have to compare the 3D structure of humanized single chain FV against mouse single chain FV is to guarantee that the structure of the canonical shape of the CDR loop is still the same as mouse single chain FV. So this is very important. As I told you in the beginning that the conformational structure of CDR1, 2, and 3, they have to come closer and stay in the very unique structure to interact with the target epitopes. So this is how we start to humanize the antibody. If you are lucky enough to have the X-ray crystal structures of the mouse single chain FV, the precision of finding the good framework is higher than by prediction, of course. But anyway, in my work, I don't have the facility to perform X-ray or CRYO-EM. That's why I am here to, it's a hidden agenda to find the collaboration with BRIN to help me find the real structure of the humanized antibody for further engineer it to increase the affinity of the antibody. So anyway, after grafting the CDR to the human framework, it doesn't guarantee that you will have a good humanist score or good CDR loops, because grafting is by prediction. So maybe the framework is not matched with your CDR. So you fail, even though you can construct the humanized single chain FV and it has no activity at all. Yes? Hello? Okay. So this is how it works. So after humanization, to be a humanized antibody, the structure of the CDR loops should be maintained as the mouse single chain FV parental antibody. So what I would like to focus today is the triple negative breast cancer. We would like to produce humanized antibody for treatment of the triple negative breast cancer. That is the most aggressive type of the breast cancer because it lacks the receptors of hormones like estrogen, progesterone, and also lack the HER2 growth factor receptors on the surface. This makes it hard to treat by the hormone analog or the available monoclonal antibody, humanized antibody, trastuzumab, that specifically bind to the HER2. So, because they are triple negative cells, so we have to find the new biological markers to treat this breast cancer. And in my case, I chose CD147 molecules. This is the CD147 molecules. It looks like it is in the immunoglobulin superfamily. So we are focusing on the domain 1 because domain 1 is the functional domain of the CD147, and triggering of the CD147 via the domain 1 change the property of the cells from normal to the cancer, or from the cancer to the more aggressive cancer. So if you can target this molecule, then you can build up the humanized antibody for therapeutic usage. My former work starting in 2008, after my former boss, have a clone of the monoclonal antibodies against CD147 in 2006. And after I finished my PhD, I come back and collaborate with Professor Vachala to produce single chain FV from the hybridoma. But this is mouse. We can show the activity of the single chain FV from mouse cloning many ways, in many ways, but it's not for therapeutic usage. So later, after I successfully produced single chain FV from mouse, I stopped considering to use this single chain FV for a long time. 10 years later, I did not work on CD147 because the knowledge of CD147 at that time is not that impressive to to produce it for therapeutic usage. But later, for the last 10 years, people describe about the function of CD147 involving in the aggressive of or the poor prognosis of tumor. And as I said, triggering of domain 1 of CD147 can cause the cancer to be more aggressive. So because it involved in invasion, angiogenesis, migration, invasion, and also even drug resistance of the cancer cells. So I come back recently, a couple years ago, I start to bring back my knowledge about the single chain FB against CD147 because trastuzumab cannot treat the triple negative breast cancer that has no expression of the HER2 molecules. So later we construct the humanized antibody by the process I described earlier and publish a paper about the humanized single chain variable fragment against the functional epitope. This antibody is unique. We are not the first one that developed the humanized antibody or fully human antibody against CD147, but the clone number M61B9 here, the clone number M61B9 here react with the special epitope that never been described before. So in this paper, I describe or elucidate the epitope recognized by single chain, humanized single chain FB against domain 1 of CD147. So this is a comparison between the mouse. The first line is from mouse amino acid sequence of the light chain comparing to the graft humanized antibody. So as you can see, the certain area that has to be kept from the mouse is, for example, this SSY something here, we cannot change because it is the CDR of the antibody transfer from mouse or graft from mouse to human framework. But certain number of mutation, it means that we would like to reduce the immunogenicity of mouse, so we have to use the human framework. So we do it from human, from heavy chain and light chain, and we got the conformational structure by prediction. The mouse and human, after prediction, we shows in the paper that they are closely similar. So it means that the loop CDR1, 2, 3, the canonical shape of these loops still looks the same as mouse. Next, just only knowing the CDR loops sequence is not that good enough for moving forward because we need to know how the complex molecules of the single chain FV and the target molecule looks like. Firstly, you have to do the docking. The docking process is also hit and miss. You cannot be sure that the docking, the post here, as I show you in the publication, is the correct one or not. Luckily that I have the collaborator who have the phase display peptide technology. She maps me the epitopes of the single chain of the CD147 that react with my single chain FVM61B9, the humanized one, which is EDLGS. So the EDLGS is the amino acid sequence that's very important in reacting with single chain FV. After knowing this EDLGS, I search for the process. So if you use the prediction by class pro docking, it will generate about 50 structures, and you have to dig up one by one, finding the one that shows the interaction and code that the EDLGS is there for the complex structures that interact with the your single chain FV. So without phase display peptide, you have to guess for 50 structures, which is impossible. So at last, I dig up one structure that shows the interaction of my single chain FV against the epitope EDLGS of the domain 1 of the CD147. After that, you have to characterize the specific determining residues, or SDR. So SDR is deep down to the CDR. There are some or certain amino acids labeled in red of the light and heavy chain that play the crucial roles in interacting with the EDLGS epitopes. The rest, they are in the CDR but have no function in interacting with the EDLGS epitopes. So after learning the red colors alphabets here, I can move further to improve the binding affinity of the antibody. Currently, the Y-type single chain FV M61B9 have the binding affinity of 10 to the minus 8 molar. So I would like to reach 10 to the minus 9 or 10 to the minus 12 to improve the binding affinity against the target molecule. Because most likely, if you would like to apply it by injecting the antibody and hope that it will target specifically target the cancer cells that upregulate or overexpress the CD147, the affinity should be higher than 10 to the minus 8. So after that, to engineer this hotspot, the red characters here, I have to use AI for predicting the specific amino acid to change in these hotspots. So the AI is by using the input sequence and filter the specific amino acids and then generate the possibility of the amino acid to change and hope that it may improve the binding affinity of the single chain FV. So I use in this work, I use the function of MCS-MAB2, which is the machine learning developed by Yu Shang-myung in the couple years ago. So, but there are a few number of publications that use his prediction for improving the affinity of the antibody. So I decided to compare the MCS-MAB2 for generating the mutant of antibody and compare it with the MD simulation. So as I told you, we focus only on the red characters to engineer it to improve the affinity. The information comes from the machine learning. So here is the prediction of the machine learning. We predict about 6 amino acids in a row. So from the light chain, heavy chain CDRs, the critical amino acids, the red characters in the CDR, in the position, for example, for the position number 32, changing from the N to Y. And it improves the binding affinity by delta T. The positive number means that after changing from N to Y, it improves the binding affinity by prediction. And we found that the most promising one from the prediction is G50Y. And the prediction by MD simulations, delta G is quite consensus with the prediction of the machine learning. Anyway, when performing the experimental work, it doesn't go in that way. Focusing on the G50Y, G50Y from this slide, it is the most promising one because it increased the delta G a lot. But in reality, when I perform ELISA, there is no binding of G50Y at all. So it means that maybe the structure that we prepare, the complex structure is not that precise, or maybe the training data set of the MCS MAP2 is not much enough to be more precise in predicting the binding activity of the antibody after mutation. But anyway, we got one clone N32Y, which can increase the binding activity in ELISA and also in biosensor. Biolayer interferometry is the biosensor to define the binding affinity of the antibody or any interaction molecules. So the lowest number is from the N32Y. And when you go back to the prediction, N32Y is not that bad. So it's still in the positive prediction. Comparing to S52Y and R96A, these two is the negative control of the system. I use it, and the prediction is the negative prediction. So it means that the binding activity should be less than the Y type one. And from the lab work in ELISA, it also show no binding activity at all after mutation from S52Y and R96A. Interestingly, although the binding activity is increased in N32Y, but when you perform the flow cytometry against the cancer cells, so this is the interaction of the single chain FB, humanized single chain FB1 M61B9. After mutation of N32Y, the population, positive population, reduced from Y type and also from the I51M. So it means that even though you have the BRI prediction, BRI affinity greater than the Y type, but when you perform flow cytometry, it's not going the same way. This may be because the flexibility of the molecule, the adaptation of the molecule in the flow cytometry experiment is different from the immobilized target molecule on the sensors. So this, I learned from this case that even though you have the good or improved binding activity, but in reality, and you would like to apply it for treatment, you have to be careful because the status of the molecule, they are different. So these are the factors that I mentioned earlier that we may encounter that the complex that we predict it's not that precise, or the training set of the machine learning is not good enough because of the data set is too few. So I think that I am reaching the 40 minutes talk, and maybe I have no time for the other molecular scaffolds, but just only 5 minutes to show you that not only the antibodies that can be built as a binder, we can also use other scaffolds. This is the list of scaffolds that can be constructed as a binder to interact with your target molecules. This binder is better than antibody because the framework structure is very rigid. Changing the amino acid will change the specificity of this binder, but it will not change the overall structure of these binders. In comparison to the loop of the antibody, once you engineer it, changing the amino acid, just only one single amino acid, as I show you in the latter slide, that just only one single amino acid, it changed everything. The binding activity changed. But this binder, other scaffolds like ankyrin and zinc finger, I use it for HIV treatment, and it can interfere the assembly process of the virus, and even interfere the integration process of the virus by zinc finger. Zinc finger is the protein that targets the DNA specifically by sequence. So I designed the zinc finger to interact with the DNA sequence of the HIV to prevent the integration process of the virus. So maybe I running out of time. So thank you for your attention. So I
thank
leave it
you.
back. Yeah.
Yeah, thank you very much, Professor Chachai, for very nice and very interesting talks. And we wait for the discussion in the end of the session. Okay, and then next we will have a second presenters, Dr. Yudi, and Dr. Yudi will. Okay, I will share the information about the title. Yeah, Dr. Yudi will deliver the presentation with the title Understanding the Virus Inhibitor Mechanism using CRYO-EM Structure Determination. So Dr. Yudi is from the Research Center for Eichmann Molecular Biology BRIN. I will read his curriculum vitae. He is a structural biologist and engaged in the manipulation of molecular cloning within diverse cellular systems, including mammalian, insect cells, and E. coli expression system. Now, he is graduate from NARA Institute of Science and Technology, PhD in structural biology, and now he is a postdoctoral scientist working on membrane protein structure determination using CRYO-EM and X-ray crystallography in University of Pavia, Italia, in Department of Molecular Medicine. I think Dr. Yudi is experienced in the structural biology and also in the CRYO-EM and X-ray crystallography. And I think this is a very important talk delivered by him about the nanobody mechanism, yeah, understanding of nanobody virus inhibitor mechanism. So, Dr. Yudi, please, time is yours.
Yeah. Assalamualaikum warahmatullahi wabarakatuh. Thank you, Prof. Dede, for the time, and thank you, Prof. Chachai. Your explanation is very nice and very clear. And I think the slide now I will share you. It's also related to what Prof. Chachai already talked about. It is related to the immunology, but I'm working with the nanobody. And also, I would like to thank you for the support here. I think I saw mostly the attendee from my research center, Egmont, and most of them may be working with the research center, our research center working on molecular biology too. So let me share my slide. So today I would like to share you my experience working how structural biology can make us understand clear about how the mechanism in molecular biology, even like in the atomic resolution. Not only I just like I said, would like to share you that our facility that provided by our government that established already for 1 year, working with the CRYO-EM, even like we have a CRYOS generation 4 that now already run mostly like almost like a 3 structure determination now. But what I would like to share you is not only about the SARS-CoV-2 because it's like already everyone already know about it, but here I would like to share how the CRYO-EM determination can be utilized for making nanobody and make a better understanding of this. Previously, I'm a postdoctoral before I joined BRIN. I'm a postdoctoral in CRYO. It is like a research center in Madrid under the Ministry of Health. And we got a funding from BBVA, one of the bank in in Spain. And also we have a grant from Diamond in Oxford and also Alba in Synchrotron in Barcelona. So I still put here my previous affiliation, even though now, starting March 1st, I joined University of Pavia. And also I have a It's one of the oldest university in the world to work with the glass not the cryos, also in the CRYO-EM facility in our research center. So I think the beginning of slide, I think not really interesting because all of this is working with the SARS-CoV-2. It is just refreshment, as we know that narrative of pandemic starting with the thing that we don't know yet what is the disease at the time. We call it disease X, and it is also we learn from this. So that's why people now already established the grant that disease X preparedness and also the pandemic preparedness and so on. But in the beginning of that time, there is no one understand what the virus is of this the pandemic. So the first research conduct data of SARS-CoV-2, there is another one, is this publication that submit in 10th of January. So this publication is a starting point for us to make the screening test, even though at the time we don't know, but after we know the nucleotide, we will understand more about this. So this publication, I mean the gene bank database is the starting point of how we make the screening test at the time. So for the genome structure of SARS-CoV-2, actually it's beginning we identify, oh, this is a virus we know and understand with another SARS-CoV-1. So it is SARS-CoV that found at that time already. So the other question is why this SARS-CoV-1 can jump to another host that is human. So this is from the electron microscopy data into 2011. This is not SARS-CoV-2, but it is the original, the SARS-CoV-1. We call it a SARS-CoV only. And this the mutation of the spike protein of this SARS-CoV can making the host jumping to the human. So even in the beginning of the key amino acid residue is only only one data change, but it is like a snowball. There is so many mutation that very strongly bind to AC2 receptor than in human. Maybe it is like the common knowledge of the SARS-CoV-2. I would like to know. I was not share you about this more. But in this structural study also can make us understand where is this virus come from. So there is another hypothesis that where is this virus come from, and also there is like a collaboration in the world that people do the sequencing and then put their data in GSAID at the time, and also
Kemudiannya pakai filtrasi berarti.
also including
Karena belum ada kan, ada yang pakai
also including the SARS-CoV-2 genome from Indonesia that originally from Litbangkes at that time, and also from Ekman Institute before joining the BRIN. And also we also here our teacher, our researcher, primary researcher submit this data very nice. At the time, it's not as much another country, but Indonesia start to submit the GSAID. So this data is very valuable data that make us understand where is the our SARS-CoV-2 is. And also at the time also we submit the data, also our team in to GSAID. So this data can make us understand not only the where it's come from, but also the strain of that in Indonesia, circulating in Indonesia. So also this is the spike host receptor jumping, how they know it, how it is from the bat, or also the pangolin in Guangdong. It is also from how they see the nucleotide that do the sequence coming from the sequencing. So by now it's like the genome structure already well established for SARS-CoV-2 because it is the pandemic. It is also influenced geopolitical and also economical issue. So there is so many grants, including the good thing of the positive thing of this pandemic, we got a huge trend at the time. So we now understand the genome structure to the protein structure always related because the protein structure is coming from the genomic structure. So and as a structural biologist, we see that our enemy is invisible, but we can like fight them using the CRYO-EM at the time is to understand how it's work. It's like a watch, automatic watch, and then we understand how the machinery coming from, and then how this movement interact to another movement. It's also analogical thinking about how we understand our enemy at the time. So we do the crystallization actually, but at the time actually crystallography is very long process. If we compare with CRYO-EM, maybe Prof. Chat say more than me because it is like a very tough work. For example, like I have a example. I got a PhD for 3 years. I only can do determination for 2 protein only. And I come back to Indonesia on March last year. We already have a 3 structure. It's within like a 3 months. So it is like a very different. So we do the X-ray crystallography, but also we do the CRYO-EM here. And then after understanding the enemy, the researcher from all the world together published the structure of their masonry system for in SARS-CoV-2. So now we now understand how the mechanism of interaction and also how do the virus life cycle of SARS-CoV-2 in our cell in the host. So this understanding, the main point is entrance point when the SARS-CoV-2 interact with the ACE2 receptor is the main point of how we can do the fighting or war of this SARS-CoV-2. It is just even like I taught you about tell you about the SARS-CoV-2. Our main idea is not only that, but also we want to see how we can make this nanobody, and the second one, how we do determination of this nanobody. So this is the entrance point. We'd like to say that the main idea is IC2 receptor and interact with the spike of a spike of SARS-CoV-2. So as we know that the 3 major treatment of COVID-19, first, it is a very global, well-known, is vaccine. The second one is compound, but the compound is only like trying to inhibit and also to reduce the virulence of the SARS-CoV-2. But vaccine is still #1. And the third one, we know it's antibody. It's also not only for the SARS-CoV-2, but also like implement to another diseases of related to the bacteria and also the virus. So we will not talk more about the vaccine and of compound, but the third one is antibody. We would like to share you that at the time we have already the cocktail against SARS-CoV-2. The idea is when the patient is already got the COVID-19, we cannot induce at the time with the vaccine because they already have, I think they have another scheme. So they have to put some antibody to reduce the cycle of the COVID-19 in their body. So there is a cocktail against SARS-CoV-2. As Prof. Chatsai said before, there is also another sophisticated and more advanced of the antibody. We call it nanobody. It is like the differences between this. We just take another part of the antibody with another part that very big and very like not functional in the treatment. So we just take another block or to work as good as antibody. So also there is a neutralizing antibody that also published at the time in 2021. There is so many and all the candidate. But the thing we know we understand that the mutational rate of the SARS-CoV-2 is very high at the time. So they make it, for example, they make it for a month, but at the time we have already new variant of the SARS-CoV-2. So the better understanding this, we already know the fragment that already established very well by another researcher in the world, not our group. But at the time, we know that this kind of the fragment can bind directly to SARS-CoV-2. It is good idea, so we will make this construct and the green construct that block the interaction between SARS-CoV and then ACE2 receptor of a human. So we design based on the structural data, we design of this fragment, both of three, because we know that there is a trimerization of the spike. Actually, spike is a trimer, and we make both of them two regions. So it is like about a 9 times higher blocking. So this platform actually try to block this in between ACE2 and spike 2, and we call it a spike body development, and we do the structural determination with our group. We submit this grant, and then we got the grant, and then we continue working with this. So there is a trimerization spike body neutralizing antibody advantage compared to the monoclonal antibody because it is very small, and also there is we can do the clone in the beginning of outbreak because we already understand how to make it. In the future, we just make the mimic to the receptor, and also we just do the cloning, and then we can start in the beginning of epidemic outbreak. And also we know that it's small, just the lack a constant region of FC and the antibody compared to common antibody, and also the more efficient because we only have take the part of antibody that block of the interaction. And for that one, we already have first we have one because we have a trimer at the time. We have to design first of the monomer one. We call it R and G. It is red and green, and our dream is to cover pan coronavirus. It's not only SARS-CoV-2 but also all this virus of the corona. First, we identify the conserved region of the spike blocking, because without this information, we just wasting the time because they make a variant so much and then so fast, and we have to understand the conserved region. So we do the analysis of data of this is a spike that conserved the region, and we got the data of this the region that very conserved at the time, and as the interaction site of the nanobody we can do. So after that, we design this one to block this all the thing. In the trimer we can see here, and then this is the monomer site. So we block this red and green, and it is just in the monomer, and after that we block 3 times. So we design the nanobody construct of this. We only take the region, and then we make the trimerization of this because our spike, as we know, is CoV2 spike is a trimerization. So we make it 3 times with the collagen. So we already established the new strategy to understanding the mutation of all the variant that come up from this COVID-19 disease. And then after that, we do the trimerization using collagen, this one interaction, but we make it producing with the cloning. We call it the arm 1, arm 2, arm 3. So this is a collagen 3 trimerization domain. Actually, this is what new of this research that from another nanobody. So as we know that the common infrastructure, we do the protein crystallography also and protein engineering. So I do it is we at the time we already just buy this plasmid of SARS-CoV-2 spike RBD and just grow it and 30 milliliter. And actually it is very difficult to establish the protocol, but our team is the first that establishing the protocol of how we have a high yield of the pure and folded protein of a spike. So we do the expression system using the cell HEC293, and then we do aspecting using purification using the cobalt. So this is data of cobalt histone from Takara. We do the gradient purification, we got it, and then after that we need to polish this one with the size exclusion chromatography SuperDeck 75, and after that we got it very clear and very clean of the protein, the target of SARS-CoV-2 spike, and we also do the thermal to see the stability of this protein is very stable, and we got very a lot 12.05 milligram per milliliter at the time. Even like the concentration high, it is very small. It's only 50 microliter. And we do the nanobody spec like first nanobody 1 and nanobody 2. To work with this, we do the design of construct, and then we do the digital trap. It is because we need a lot of the plasmid data at the time. We do the culture of amplification of plasmid even in 4 liter. So we do the giga trap at the time. We put all of this, and we got a lot of the plasmid. So this is our plasmid, and then we do express with hecton entry again, and then we do the isolation of within strep tag. So our purification done in with this, we directly the no gradient. We just got it, and then we do the polishing with the SuperDex, and we got very pure protein this time. And even we got like a 5.6 milligram per milliliter, 130 microliter. And then we do the thermoflow for stability assessment. And we found it a very nice thermoflow analysis. And then we also try to see the spike of protein purification quality control. And then we see very nice also here. And the result of this study is to understand how it's like to see the understanding in the different mutation spike of SARS-CoV-2, and also the second one, how much the concentration needed in the beginning. So we do the neutralization assay. As we know this data, it is the nanobody 1, even like nanobody 2 and nanobody 1 is can inhibit of the interaction between SARS-CoV many variant and also the IC2 of receptor of human. And this is the main point of the idea is we already have this data of a cryo-EM. So we take this protein and we incubate for 2 hours, and then we put in the cryo-EM facility in Madrid, and then we determine the structure. So the structural determination make us understand where's the binding in, and how we can make a better nanobody in the future. So we do as usual as what we did in CRYO-EM facility in BRIN. We just take the micrograph and then do the molecular replacement, and then we do the data processing at the time, and the final is the validation. This is actually the recommendation method what we got using Reliant. Even like in BRIN, we use a more sophisticated one is Cryos, and also we use a Cryospa. Here in the beginning we use a glass phase. The voltage is 200 kilovoltage. Even like this is Talos in BRIN, I think it's better. It is in the voltage, and also the camera it's using Gatan K3. In BRIN we have serta and also Falcon, the best one. Falcon G4, I think that is the best one. And we got this data from 163,000 particle of this. We do the screening of this. I think most of team of the CRYO-EM facility understand about this CRYO-EM data. So the idea is we see this one, this picture. It is 2D picture, right? After 2D picture, they combine 1,000 of data to be 3D structure, and this 3D structure we will analyze with a refinement and so on. And after that, we got the 3-dimensional structure of this. So after a long time, we only got 160,000 particles. It is like a very small amount. For our experience in BRIN, we took about like a million of the particle. But here we just only 160,000. But we already have the good resolution, 2 angstrom. If we see the size of protein is very big, so it is acceptable resolution of electron density map. After working with the reliant and filtering the 2D and 3D structure, it is so long, maybe like in my experience working with this, maybe like a 1.5 month at the time. In CRYO-Spark and BRIN, we just maybe like several days, we already got this data. I think it is a most sophisticated one. So we got this data. As we can see here, the coloring, this one is it's a spike protein, but the other one, the upside, it is our nanobody that interact directly to spike. So it is also we can move this one and as we say a top view. So this is data is very valuable to us to understand how they interact. And this we can see this is RBD, and then also this is the nanobody E and also nanobody V. So this is how they interact in each other. If we see in top view, this all is yes, it is as our hypothesis. This interact with the conserved region of a spike. So that's why our nanobody is more potent than another nanobody at the time. So this is also we can see as easier as how the interactions is. So the RBD and then also the nanobody interaction. That's all. But I think I think it's impossible to share you about how this interaction is because I think it's too deep. I think and also we already published in Advanced Science last year. You can download the data of that. So also this the structural study of we call it 3-dual blocking of this protein. That's all. This is our lab facility. It's like a 15 minutes from Santiago Bernabeu in Madrid, and then now I moved to Italy in the nearby 20 minutes from Milan. So thank you for listening, and let's discuss together about what is a CRYO-EM is. Thank you.
Okay, thank you very much, Dr. Yudi, for a very nice presentation about the nanobody inhibitor mechanism for COVID. And now we invite to all the audience question. And please, if you have any question, you can raise hand and open your mic, or you can also write your question in the chat room. Is there any question to the
So I have a question for Dr. Yudi. So
okay, that's right.
yeah,
Please.
your nanobody bind to the open conformational structure of RBD, right? Or closed structures? It bind to the open structures of RBD or closed structure of RBD.
Yeah, actually, we have two nanobody. The nanobody one is we see it's a bind to closed structure, and the nanobody two is a bind to the open structure, and we see in between also. Yeah.
Good.
Thank you, Prof.
So, Dr. Yudi, you use these two kind of nanobody, right?
Yes.
Just to compare, or
Yeah.
you
This
compare with this?
yeah, the nanobody
Okay.
one is actually it is very different in the kind of the amino acid residue that I cannot because it's not published yet. But another nanobody also have another key residue with this. But the idea is yes, we want to see in the processing that Prof. Chatsai also asking about this one. But our limit is we cannot see the collagen interaction in between a trimerization domain. So now, until now, our group still working of crystallography to determine the structure of the coil one, the flexible one. So it is like a very valuable information on how they like make open structure and also the closed structure related to our nanobody that we cannot see because it is 3 point something resolution. So hopefully in the future we can publish even like it's very difficult to publish the structure of SARS-CoV-2 again because it's not very sexy anymore. But we see the potential of this. Thank you, Prof. Dede.
Maybe the one that interact with the open structure is better because the open structure is the time that the virus interact with the RBD, with the receptors, right?
Yeah.
Okay, I think it is a very simple question for the common people. We don't understand about the antibody. From your speaking, what is the difference between the monoclonal antibody and the nanobody antibody? For example, if can we use like a monoclonal antibody to in this SARS-CoV virus?
Yes, Prof. The idea is monoclonal antibody. For example, we do the production of monoclonal antibody. We induce some. There's we call it the common conventional one is a hybridoma process. So we like collect of the monoclonal antibody from the injected mice, for example, and then put in some kind of the technology. But the sophisticated one is called nanobodies. Just take the letter Prof. Cacai clearly like explain us. It is just part of the the functional of the antibody, and that can work as potent as a monoclonal antibody. So of course we can use this one for all the diseases. So the idea is the strategy is to know how the structure or the way of the nucleotide and then can block this protein interaction. So the idea is just that. Yeah, like we know that the protein is like a key and substrate. So in our idea, in key and lock this one. So if the structure is very competitive to another natural region, so it is like can block directly.
I see. Thank you, Dr. Yudi. So I invite the audience to have a question to all the speakers here, please. Okay, I have a question to Professor Cacai. So the CD147 is actually mostly already reported in the PDB, right? The structure of the CD147. I don't know the difference between the domain 1 and the domain 2, whether it is already published in PDB or not, and why the breast cancer is mostly affected in the CD147 domain 1. So, and then when we CDR grafting, how we can measure the successful of the grafting CDR in the humanized? Yeah, can you explain, Professor Cacai?
The first question is about the structures of the CD147. It's already published as X-ray crystal structures, but the antibody against domain 1 is important because domain 1, as you see from the diagram, that is at the tip of the molecules, so it's easier to access the receptors. So in the function of the CD147, once it overexpressed, it interacts with the other molecules on the surrounded cells. For examples like macrophage or monocyte. Nowadays, we know that there are good and bad macrophage. So bad macrophage have the molecules that can interact with the CD147 and induce the aggressiveness of the molecules. So that's why the D1 domain is more important than D2, and by nature, because it's at the tip of the molecules. So people knows the structures for a long time ago, but for the one who interested in production of the monoclonal antibodies, I think there are just only two structures
to complex structures of their antibodies against the CD147. And I select the one that interact with the domain 1 and pick up that domain 1 for the molecular docking using the class Pro. So because I used to use the whole molecule of CD147 that contain both D1 and D2, but the docking process is not that precise. So I then small down the structure of the molecule. Luckily that there is one group that published the interaction of their antibodies against the domain 1. So I pick the domain 1. So that narrow down the possibility of the interaction. So otherwise, if you use the whole molecule for docking, it's very hard to get the most promising one. And what is the second question? So I answered already the X-ray crystal, domain 1, and what's the next one? Oh, the success of grafting.
Okay.
The success of
Grafting.
grafting can be defined in the beginning of the process by molecular modeling to see whether it's the IMSD of the generated immunized antibody is close enough to the mouse antibody. But as I said, in my lab, we don't have the facility for X-ray crystallography. So we go by modeling in the beginning. But the modeling of the antibody is quite precise nowadays because from the machine learning, AI, it makes it more precise comparing to the last 10 years that people just use homology modeling of the CDR loop, which is not that precise. So I can rely on the prediction from the machine learning. So then the overlapping of the 3D structure can tell you in the beginning that it should work. But anyway, you have to perform the experimental work as I show you by ELISA, by biosensors, by flow cytometry to get more information about the binding activity. And from the data I showed you that the prediction of machine learning is not that precise. And maybe because our starting material is not that precise, the complex structure is not that precise. So we cannot rely on the machine learning only. So, and from my experience, even though you have the good binding activity, improve the binding activity, as you can see one clone that have better binding activity, but when you perform the flow cytometry, the binding activity is not that great comparing to the wild type. So it means that the status of your target molecules, if you immobilize on the solid phase, as in biosensor or in ELISA, is not comparable to the molecules that stay in the surface of the cells. So the flexibility and adaptation of the binding of your antibody, modified antibody, and the adaptation of your target molecule is not just lock and key, but it induces fit of the molecules. So this lesson tells me that even though you can improve the binding activity, and if you are playing with the surface molecules, you have to confirm the binding activity with the proper methods, which is not immobilized on the solid phase. Immobilized on the solid phase, your target molecule cannot move. So it's different from the interaction of your target molecule on the surface of the cells. So that's what I learned from this work. I hope
So yeah,
answer your question.
so after you confirm with the ELISA and also flow cytometry, you got this idea about the sometimes our antibody immobilized in the surface, and I think measuring the binding site.
Yeah.
Okay, final question for Chachai. What is the size of your antibody? I mean, crystallography is good for the very low molecular weight, but the CRYO-EM has a limitation about this. So I asked about how many size of your antibody size.
Yeah, the antibody size is around 15 because it's just only the short form, single chain FV, 15 to 20 something. So I already discussed with Dr. Yudi that maybe we cannot guarantee the success of that small size. But I hope that once it interacts with the target molecule, which is larger, so the combination size of the target molecules and the single chain FV itself, it may be improve the precision of the CRYO-EM. So including the target molecule, it should up to about 50 kilodalton, 50 kilodalton. That may help to visualize the structure in CRYO-EM. But anyway, we can go by both ways for crystal structures. And I think crystallization, as you mentioned earlier, that as I asked you earlier, that people can crystallize CD147 and also crystallize the single chain FV. So we can follow that process to perform the X-ray crystallography in the beginning of our collaboration.
Yeah. We hope we can analyze when we use the antibody and the target cells, and then you said that the size is around 50. Then we hope that we can see in the CRYO-EM facility.
Yeah.
Dr. Yudi, have you have any comment about this?
Yeah, I just want to refresh that we have because there is the attendees also from Indonesia. We have a program, Prof, that we open platform for the CRYO facility and also the grant starting this week. I think we open the proposal of this, and many of you I think have already put ICP the idea, and then we can do the collaboration together. Even like I'm here in Milan, I hope I can contribute with this.
I have a
Thank you, Prof.
framework of the project. I have the framework of the project already. So just only add on the X-ray crystallography and CRYO-EM parts. We should get the grant.
Yes, thank you very much. So before I close, there is a question in the chat room. I will read. I don't know, Professor Chachai or Dr. Yudi can answer this question. Good morning, everyone. Very nice presentation. Structural biology is very new for me, especially CRYO-EM. As we know, we can find the structure of a protein or compound as a result of CRYO-EM. Can it be used directly for in silico molecular docking? We usually use compound or protein structure from database for in silico analysis. And then how is the plant sample preparation process for CRYO-EM? Is it different for cell or tissues? I don't know who, Prof. Chachai or Dr. Yudi maybe can answer the question.
I just want to say this one. It's what I did in my experience. Structural biology is very new for me. Yeah, it is also for us because it is like a new technology, right? So don't worry. And then as we know, the final structure of protein component, yes, it can be determine the structure of the protein and also the compound as even like this compound is very small. Usually like we put the artifact inside and then put the refinement with this. It's very difficult thing to see the compound in the CRYO-EM. Can be directly for yes. Actually, in silico, usually people working with this in silico protein is coming from the protein data bank that also coming from even like there is three mostly X-ray crystallography, CRYO-EM, and NMR. So these three things, it is the I want to say that in Indonesia, Prof, many people working with the in silico processing, in silico analysis with docking, with molecular dynamic, and so on. And they like sometimes they confuse with what is different between our working with the CRYO-EM and what they did with the molecular dynamic and also docking. So I want to answer this confuse that molecular dynamic and also molecular auto docking or also docking and in silico analysis. It is utilize the data is coming from us, from the CRYO-EM or from X-ray crystallography. So the primary data, it is from the structural biology experimental. So it is because Prof Dede now that in Indonesia, like, oh, CRYO-EM is like in silico. It's very different. It's a very different concept. So yeah, we usually use a compound of protein structure from database for in silico. Yes, it is from that PDB. And then how is the plant sample preparation process for CRYO-EM? Actually, I don't have experience, so I cannot answer this. So it is my limitation. I'm only working with the mammalian, also human. But my concept is maybe if Mbak Intani already have now the gene and also the protein target, you just can clone to, for example, another expression system. But I don't know where's the clause of the plant protein expression. Maybe like a yeast or something. I don't know. But we need to analyze this because we don't understand about the plant sample or plant gene. But yes, if we already produce so many protein and expression system and don't purify, we can do the CRYO-EM. And on the other hand, I think additionally we can do the architecture of cell using Aquilos that we also have with the plant. But I think it is very challenging to prepare the very tiny and also thin the thickness of the cell of the plant. But I think in the future we would like to establish also the Aquilos facility in BRIN. Yeah, maybe that, Prof. Dede.
Okay, thank you very much, Dr. Yudi. I will add some information about the CRYO-EM. That this question also I already asked to Dr. Pan when the workshop last year about the whether CRYO-EM facility can analyze the chemical structure of the, for example, plant extract. The answer is very difficult because you can use NMR to analyze the structure of the plant chemical compound, but because the CRYO-EM has a limitation for the size of the protein. But if you want to analyze the protein target for your compound, then you can analyze the protein target structure. Then you can predict, not predict, you can see that your compound can interact with some part of the amino acid in your protein. Yeah, you can analyze it using the CRYO-EM. I think this is the answer and the addition from you. Okay, before we close, is there any question or comment? One question is okay. If no, so we are very thank you to Professor Chachai and also Dr. Yudi for the very nice presentation. And this is very related to how we develop antibody for our disease, for example. And then we hope that we can make a very good collaboration with Professor Chachai. And this is information for all the audience. If you have any potential collaboration with the Professor Chachai also, you can make a collaboration and you can really use CRYO-EM facility to make this collaboration. I think we hope that our research on the CRYO-EM facilities and structural biology in the future will be more successful. Thank you very much again, Professor Chachai and Dr. Yudi, and I will give this to Mbak Andalusi. Mbak Andalusi, please. Thank you very much.
Okay, thank you, Dr. Dede, for leading a very interactive discussion. And thank you to all speakers for a very interesting presentation. Ladies and gentlemen, this is the end of our agenda for today. I'm Andalusi Arini. On behalf of the committee, I would like to say thank you very much to our speakers, moderator, and all participants. See you in the next event. Wassalamualaikum warahmatullahi wabarakatuh. Thank you.
Thank you very much, Professor Chachai. Yeah, we can make more email for next discussion.
Okay.
And also Dr. Yudi, masih jam 5 pagi ya?
Iya, jam 5.30 sekarang.
Thank you, Professor Chachai.
Terima kasih, Prof. Thank you, Prof.